And then we're going to come and tell you a little bit about what we've been doing in our laboratory, the aims for
So first of all, I'd just like to thank a variety of people very quickly for collecting the data and analyzing the data, these individuals in my laboratory. To thank Dr. Anjay Barkey for the colony several years ago after I left my postdoc, and for some individuals at our USDA Human Nutrition Center who helped with the metabolism work along the way. And of course to thank the support of the NIH primarily, and the Glenn Foundation and the Ellison for their help supporting the work throughout the last several years. So Dr. Barkey introduced the Ames dwarf mouse to you yesterday very nicely, so I will cover that fairly quickly. So again, this animal is growth hormone, prolactin, and TSH deficient, and their lifespans increase by about 50 to 70%, males and females respectively. Their antioxidant defense mechanisms are enhanced, and I'll go over that in a little bit more detail in the next slide. And we know that if we provide growth hormone, both in vivo and in vitro, we can downregulate some of those enhancements that I'll talk about in just a second too. We know that thyroidoxin and glutaredoxin levels message protein activities are increased in this animal, and they have an enhanced resistance to both in vitro and in vivo cellular type stressors. So this is just a summary of the oxidative metabolism in the mouse. We showed that mitochondrial hydrogen peroxide production is lower from liver mitochondria. We know that catalase, SOD, glutathione peroxidase are all significantly elevated. We've looked at message levels, protein levels, and activity levels of these enzymes across various ages. We also know, as I mentioned a second ago, is if you provide growth hormone in vivo or in vitro, you can downregulate each of these antioxidant enzymes. We also know that non-enzymatic antioxidants, such as ascorbate, glutathione, and methylthionine also differ in these animals. And what I'm going to focus the rest of the talk on is glutathione primarily, and how that leads into methionine and some other changes with methylation. This enhanced antioxidant defense in the Ames dwarf is functionally significant too, because if the animals are provided an in vivo stressor, they're able to overcome that stress better than a wild type animal, and this work's been done by other individuals. We know that accumulation of oxidative protein damage, DNA damage, and some damage to lipid via lipid peroxidation is also suppressed in these or significantly reduced in these animals over time. And we know from the mitochondrial work that a variety of the oxidative phosphorylation enzymes are upregulated. And again, this is activities, message expression, that type of thing. Okay. So the glutathione levels were intriguing. We made this observation several years ago, showing that liver glutathione levels are enhanced in the Ames dwarf across their lifespan and probably beyond. We only measure things typically till 24 months, because that's when the wild type animals tend to start dying. But we also measured the oxidized form of glutathione, the GSSG, and it tends to be much higher. So the total pool of glutathione is higher in these animals overall when compared to wild type. So we looked further into glutathione metabolism to determine why there would be differences in the total levels. And we were able to show that the rate-limiting step in glutathione biosynthesis, the glutamylcysteine ligase, that enzyme is significantly elevated. The levels of glutathione as transferase, which we consider an enzyme that utilizes glutathione, those levels are significantly elevated too, which contribute to detoxification differences in these animals. And an enzyme called gamma-glutamyl transpeptidase, which breaks down glutathione, is significantly lower in these animals. And so we think that the total, all the differences in biosynthesis, an increase in biosynthesis and a decrease in degradation, help contribute to the elevated levels. When discussing this data several years ago with an individual biochemist at at our Human Nutrition Center on campus, he suggested that we look at methionine levels and the whole methionine metabolic pathway. So we followed through with that work and found that the entire methionine pathway is altered in the Ames dwarf mouse in comparison to the wild type mouse. And this is what leads to these higher levels of glutathione. So specifically, we saw increases in the level of the enzymes that contribute to this metabolic pathway. Again, we've looked at message levels, protein levels, and the activity levels of these enzymes, and they're elevated 50 to 100% in the Ames dwarf compared to the wild type. And then if you add, again, growth hormone back into the system again, so administer it in vivo, you suppress the activities of these enzymes, suggesting that growth hormone is involved in regulating this particular pathway. Let me see here. This is just another way to show you these increases. So again, this represents message expression with the wild type animals are representing a level of 1. So these are increase or upregulation, fold increases in the amount of message expression in the Ames dwarf compared to the wild type animals. And you can see that overall, that there's a significant upregulation with the exception of one particular message. Let me go back here. So methionine can be processed or follows a pathway down to homocysteine, which can either follow two pathways. One pathway, this is called transsulfuration, and this pathway is a recycling of the homocysteine back to methionine. And the only enzyme that we found a decrease in is methionine synthase, which suggests that the pathway is driven this direction. And I'll show you a little bit of other data with that in just a second. So anyway, so this is the only enzyme that's, the only gene that's downregulated in the dwarf is that MTR, methionine synthase. Dr. Barkey also mentioned a growth hormone transgenic animal, I think, a couple of times yesterday and maybe this morning. These animals were produced to overexpress bovine growth hormone, and they live half the lifespan, so they live 12 months. And so we had the two extremes, the Ames dwarf that's devoid of growth hormone and the growth hormone transgenic, which has very high levels. And so we looked at methionine, the expression of these methionine genes in the growth hormone transgenic animals also, and we can see that they're, for the most part, significantly suppressed. So here's one, which represents the levels in a wild type animal, and the levels in the growth hormone transgenic are suppressed in comparison, with the exception of one particular enzyme that's involved in methionine in the folate pathway. Again, if you give growth hormone to these to the dwarf mice, or if you look at these growth hormone transgenic animals, you see a suppression in this pathway, again suggesting that growth hormone may regulate methionine metabolism. So what I've described in the first few slides is that the Ames dwarf has differences in glutathione levels, in total glutathione levels, and that we think the contributing factor to that is this altered methionine metabolism. So the altered methionine metabolism leads to an increase in cysteine concentrations, which are driven through this transsulfuration pathway that I mentioned to you, and that's what helps raise the levels of glutathione. But there's another pathway that's also involved in the methionine pathway, and that's DNA methylation. So let me see here, let me back up right here. In this step here, the S-adenosyl methionine to S-adenosyl homocysteine, there's an enzyme called glycine N-methyltransferase, and this methyltransferase is really highly upregulated in these animals. And we know from the literature that the SAM to S-adenosyl homocysteine ratio is supposed to be very important with regard to metabolic function. Let me bring this through. And so the literature again suggests that when methionine is abundant, that the transsulfuration pathway is favored. And when methionine is low, that transmethylation is favored. And so a further study we did was to look at the flux of methionine through those pathways in the dwarf and the wild type to see if there was a difference in not just the enzyme levels and the activities, but to see if there was a difference in flux. And what we did find is that the methyl moiety and carbon chain So methylation and transsulfuration are significantly increased 2 to 3 times in the Ames dwarf in comparison to the wild type. So the literature doesn't necessarily apply to this particular mouse because we have both transsulfuration and transmethylation activities increased in this animal. So that we began to think more about methylation, and that SAM, or S-adenosylmethionine, is the universal DNA methyl donor, and suggesting that there was potential for DNA to be modified in these animals epigenetically in comparison to the wild type. So the first thing that we did, I had a graduate student working on this, and to look at we looked at global hypomethylation, and chose to look at interspersed repetitive DNA elements because it represents a very large portion of the DNA. And she looked at 4 different ones, and 2 of which I'll show you here. So line L1 represents about 17% of genomic DNA in the mouse. And we found that in the dwarf, in comparison to the wild type, at 3 different ages, the dwarf tended to be more hypomethylated than the wild type, which isn't exactly what we thought we would see with an increase in methylation based on our earlier data. And then the other one we looked at was sign B2, and the dwarf tended to be less hypomethylated in comparison to wild type. And then we also looked at just gene expression or message expression for line L1 and sign B2, and there weren't any differences between the dwarf and the wild type. Also, my graduate student didn't think if it's not a 0.05 significance, she was unimpressed with the data. And we tried to tell her that even 0.07 level was very significant. If you're representing 17% of all the genomic DNA, it might be really important. So she's going to follow up with some of those studies, and we want to follow up with specific gene expression or specific gene methylation data also. So a couple of years before we did the hypomethylation data, we looked at DNA methylation using some arrays, and we found that there were 100 differentially methylated genes out of about 3,000 that we looked at, and 81% of them were methylated in the dwarf. And not at all in the wild type. So now we're looking at those individual genes to look at message expression. And these are just a few that are listed because the laboratory is truly involved in stress resistance. So we found various ones that are involved in stress resistance. And one of the examples that I'll show you as far as gene expression is concerned is GSTP. The GSTs, or glutathionase transferases, I mentioned that before, they're upregulated in the Ames dwarf. And we had just looked at a single activity level at that time to evaluate GST. We've now gone back and looked at, I think we've looked at 12 now, GST isoforms. It's a huge family of enzymes. All of them are highly upregulated in the Ames dwarf, with the exception of GST pi, or P, as shown here. So, and we know that this particular GSTP is methylated in the Ames dwarf and not methylated in the wild type. And sure enough, expression is suppressed in the Ames. Now, one thing I mentioned the growth hormone transgenic earlier. So this is the animal with the very high levels of growth hormone. This particular animal, we looked at GSTP or pi message expression, and the levels are much higher. So we haven't looked at methylation yet in these animals, but it suggests that perhaps growth hormone may be regulating methylation. Again, when we see opposing expression of message for this particular gene. So we've also looked at the DNA methyltransferase enzyme. So we knew that glycine and methyltransferase was up about, it's 100% the activity is higher in the Ames dwarf compared to wild type. And when we were thinking about DNA, we decided to look at the 3 different DNMTs. It's DNMT1, 3A, and 3B. DNMT1 is primarily responsible for maintenance methylation, and 3A and 3B are primarily responsible for de novo methylation, but there's a lot of overlap we're finding in the literature anyway. So we looked at expression levels of DNMT1, 3A, and 3B. They're higher in the Ames dwarf across 3, 12, and 24 months compared to wild types. And this is in the liver. We've also looked at just an activity assay, which you can't distinguish between DNMT1 or DNMT3A, 3B activity. It measures all the methyltransferase activity, and it's higher in the Ames dwarf in comparison to the wild type. And then these are protein levels. And so this represents DNMT3A across three different age groups. Again, the levels are significantly higher, but the level of DNMT1 was suppressed, the protein. We found higher levels of the message, but we found much, much lower levels of the protein in the Ames dwarf compared to the wild type. And so we explored that a little bit further and used our in vivo study where we provided either saline injections to wild type and dwarf animals, and then we also, a third group, we had dwarf animals that we added growth hormones. We provided them with growth hormone a couple times a day, and then we removed their livers and we looked at DNMT1 expression. Sure enough, so we were able to repeat what we had seen earlier in a different group of animals, that the protein levels of DNMT1 were lower in the Ames dwarf that were treated with saline, and when you provided growth hormone to the animals, it raised DNMT protein levels up again. Okay, thanks. And then in vitro, we also took primary hepatocytes and cultured them, and when we didn't provide anything or a very, very low level of growth hormone, 0.1 micrograms, we didn't see any changes in DNMT1, but when we provided higher levels of growth hormone, DNMT1 protein levels went up. And we saw the opposite effect for DNMT3A, that the levels were lowered with the growth hormone concentrations, although we didn't really find a dose response in this case. So in the absence of growth hormone, in the case of the Ames dwarf, we have an increase in methionine metabolism. And this leads to an increase in methyltransferase activity, and we hope to see an increase in gene-specific DNA methylation. We've shown that for one gene only, but we know that the dwarf is differentially methylated, and they have elevated DNMT3A, but a suppression in DNMT1. And so we have to figure out a way to measure activity levels and potentially alter those different DNMTs. We also know the increased methionine metabolism leads to an increase in cysteine availability, which leads to an increase in thiol compounds, of which I mentioned GSH and metallothioneine, and there are others. And in the presence of growth hormone, you have a decrease in methionine metabolism, which alters methyltransferase activity and cysteine availability, and that leads to some differences in defense mechanisms in this animal. So we think that growth hormone causes a shift in resources. So when in the presence of growth hormone, there's an increased demand for amino acids for growth. And in the absence of growth hormone, such as in the dwarf mouse, there's a decreased demand for growth, of amino acids for growth. And then finally, why might methionine levels be important and epigenetics be important for aging? We know that the folate pathway, which includes choline, betaine, and the methionine and homocysteine, we know that when those levels are altered in humans and in animals, either by deficiencies or overexpression, that it can cause disease. And we know that growth hormone is really promoted as an anti-aging factor, although we know in the laboratory that growth hormone suppresses antioxidant enzymes tremendously, and it lowers defense mechanisms. It also may affect DNA methylation. So I'm on the side of anti-growth hormone for treatment. Anyway, so we think that reduced somatotropic signaling allows for metabolic reprogramming, in parentheses, or in quotes. So it shifts resources away from growth and proliferation towards stress resistance and cytoprotection. And we believe that some of the mechanisms that contribute to that involve epigenetics, and that's what we're pursuing currently. So thank you very much.
Questions?
So in the germline, methylation is completely erased, and so I'm wondering at what stage you think this actually comes into play, at least for development, right? Because even in the Ames dwarf mouse or any of the little mice, they grow for a certain amount of time, but they just ultimately don't become the same size as the adult.
It's a really good question. It's unclear. So what we're doing. It's a great question. It's unclear to us, and so what we're doing is backing up our studies. So we're currently looking at 4-week-old mice, 3-week-old, 2-week, and day of birth. The problem is they're still in the environmental system of the female. Through the pregnancy and then up to 3 weeks afterwards, they're still receiving potential hormones and things via the milk. So it's kind of uncertain at this point, but that's what we've been thinking about too.